Hey, today we're going to talk about 8.12 and 8.13, lethal dose 50%, LD50, and dose response curves. So there are two topics, and this is the only video that is going to join two topics, but they are very closely related, so this makes the most sense. Take a moment, check out the Enduring Understanding, the learning objectives for both, and review the essential knowledge as you set up your Cornell notes. So, in order to determine the lethal dose 50%, this means the amount of a substance, whether it's a chemical, whether it is any sort of pollutant, any molecule, any dosage for a drug that they're testing. This is when 50% of the individuals that are tested will be dead. So you want to figure out the lethal dose, because then from there you can figure out an effective dose, and you can work your way backwards from that. Now, for the purposes of this lesson, we really just care about the lethal dose 50%, and this might be, for example, if we wanted to figure out the effectiveness of a pesticide. Now, a couple of things about this. You're going to have your independent variable on the bottom, and that is where you'll measure your concentration, your dosage, the mass that you're adding, and the pollutant or the chemical in question will be added either to the air, water, food, etc. And on the dependent variable, the y-axis, we will be measuring the percent mortality or the number of individuals that are impacted, something to that effect, and it's a response to whatever dose we are adding. That's why it is a dose on the bottom and response on the left, so dose response curve. Now, the other weird thing about this is we use this crazy graph paper that's called semi-log graph paper. And this graph paper does not have zeros. You don't count in normal, it's logarithmic. So as you jump between these major divisions, it's a multiple of 10. So just to move closer, this is 0.0001. Notice 3 zeros. Here I have 2 zeros. Here I have 1 zero. So as we count from the left, 0.0001, then 0002, 0003, 0004, 0005, 0006, 0007, 0008, 0009, 0010. And that's exactly what we have here. So I just took off that zero at the end. Now from here, we're going to go 0.002, 003, 004, 005, 6, 7, 8, 9, 0.01. Then goes 0.02, 0.03, 0.04. Another way you can do it, you can do 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 20, 30, 40, 50, 60, 70, 80, 90, 100, 200, 300, 400, like that. So as you move to each one of these new divisions, you're increasing or multiplying by 10. And when you're finished, you should have an S-shaped curve like this. And this, if it doesn't make an S-shaped curve, you did it incorrectly, or it is not a lethal dose 50 or an LD50 curve. We are mostly concerned with this value in the middle, but we're also concerned with this value down here, because this is when we start to see the impact of our dosage. And this is called the threshold. And it's the dosage wherever the effect begins to occur. So it's wherever that curve starts. Now I did this with lines, but when you draw it out, you would use a curved line. You would use a best fit line. In this graph, you never just want to connect everything, because you might have some outliers. This, I made it so it looks perfectly, but you might have one that's like way over here or something that's way over here, and you don't want to draw it like all over the place. You want to just literally make a nice curved line. And again, that is the threshold. But when we're trying to figure out the lethal dose 50, we are going to look at the y-axis and go to the halfway mark. If it's percent mortality, that's easy. Draw a line at the 50%. If it's number of individuals, then you go to the halfway point. Either way, you want to do it halfway up on the y-axis, draw a line straight across, and then draw a line straight down. And from that, you'll be able to figure out your lethal dose 50 in concentration. So just to bring it a little bit more in focus, we have 0.01, this is 02, 03, 04, 05, 06. So I would say this is about 0.02. Sorry, 0.022. I want to be as precise as possible. Now, we're going to come back to this value right here. So this would be, again, 0.2, 0.02, 0.03, 0.04, so on. We don't ever use the LD50 curve on humans. It's not ethical because people are not just going to volunteer their life for the sake of science. We would use models. For humans or other mammals, we would use we would use mice and rats. For aquatic reptiles and amphibians, we would use fish. And for terrestrial reptiles and amphibians, we would use birds. And it is expressed in milligrams per kilogram, typically. If it is requested, you could convert your values to another value, but typically it is in milligrams per kilogram. Now I'm going to do something a little bit different. I'm doing this in grams, but let's just look at an example. The LD50 of a particular chemical is 50 milligrams per kilogram. So this is, again, the lethal dose 50. So calculate the mass of chemical in grams to elicit a lethal response in a 75 kilogram human. So what we want to do is set up a proportion. And we want to start with our original lethal dose. Then we want to upscale it to the mass of a human. And to solve this, you will, of course, cross multiply and divide, and you get 3,750 milligrams. However, my question wants the answer in grams. So now I have to do a little bit of dimensional analysis, and I will start with the 3,750 milligrams and then cancel out the milligrams because 1,000 milligrams equals 1 gram. My answer is 3.75 grams. Now, did you need to do all this dimensional analysis? No, you did not. You really just moved the decimal over because grams are larger than milligrams, and they're off by a factor of 1,000. If you knew that, great. If you didn't know that, there's no shame in showing your work and setting up the math problem. Now, lethal dose LD50 studies are typically done short term. So they're acute studies. The word acute refers to short term. We do not really know the long-term effects from these studies, and also we don't really look at the ecological impacts. So for example, if you're testing a certain pharmaceutical, like you might not know how it impacts if it is you'll know if it's persistent and organic. However, you won't know how it impacts other organisms within an ecosystem. So chronic studies are long-term studies, and that's where you would study an organism from its entire life cycle. Like, for example, studying a fish health from hatching to their reproductive or sexual maturity. Now, these, again, acute studies are used, or LD50 is used in acute studies. LD50 is not used in chronic studies. There's a lot more information that is required when you do a long-term study. Now, you're going to practice. I have the setup from before, and this is the problem that we did before. But according to the dose response curve to the left, the LD50 for a typical laboratory rat, which weighs 300 grams, is 0.22 grams. Calculate the LD50 for a 75 kilogram human. So I look forward to seeing your responses. I hope that this video was informative. Thank you for your attention. I will see you soon.